Hi, it's Alistair Ewing from AME Bioscience, and welcome to the first video interview that we have produced. Now, the purpose of these interviews is to talk to people in companies that are developing something new or an emerging technology and make people aware of these developments within our community. The first interview is with Ralph Coney, who is the Chief Operating Officer from BJS Biotechnologies. Now, this is a company that's based in the UK, and I knew of them, or have known of them, for a couple of years through their ultra-fast real-time PCR technology. However, Ralph will discuss in more detail a new product which they're developing and which is about to be introduced. And it's an integrated molecular diagnostics platform. And they have a version of it which is specific for COVID-19. So very topical in these difficult times. Now, the format, unfortunately, is not exactly as we had hoped with the video recording, but it still works. So we'll look at a different format for future interviews, but as things are just now, we'll leave this format as it is, and we'll just go on to the interview with Ralph Coney. Okay. Hi, Ralph. Thank you very much for agreeing to participate in the very first interview as part of the new AME Bioscience website.
That's fine.
I've followed your company quite a bit. I know you've got a lot of innovation, so why don't we just kick on in and tell me a little bit about your company and your products.
Okay. Well, I mean, the company has been going for 75 years. We've grown.
What's the name of the company, Ralph?
The company name is BJS Company, but I work for a subsidiary called BJS Biotechnologies. And BJS Biotechnologies has been going since 2011. And in that time, we've created an innovative, fast qPCR system. It uses a totally unique heating system. So it uses resistive heating. And the reason this was developed is because we also are the leading manufacturer of silver blocks for conventional PCR machines. And we recognize a lot of the limitations that exist with existing Peltier-based qPCR systems. So one of our engineers came up with a solution to create an ultra-fast qPCR system. That system is built and is currently being sold under the brand Express NGX. We also, in the last 18 months, have been developing fast molecular diagnostic systems. And this is in recognition of the fact that we saw there was an opening in the marketplace where you could use ultra-fast qPCR to be able to create an ultra-fast molecular diagnostic. So you would have a sample in, result out solution.
So this is a single integrated platform.
A totally integrated platform. So you would just put it into a standard tube. It's got a QR code on it. You scan it, you load it into the system. It runs completely without any human intervention, and results come out at the end. And that will run on a suite of STI, respiratory, gastroenterology. So that was our long-term aim, was to do that probably in about a 2.5 year period.
Okay.
As a result of the recent COVID-19 crisis, we've recognized the fact that we can actually produce a cut down version of that system, which can be produced very cheaply and very rapidly. We can go and manufacture that within a 6-month time frame.
What do you mean by a cut down version?
Well, basically that will just run COVID-19 tests.
Okay, and it's a fully automated.
It's a fully automated system. Okay. And once again, that would also include another innovation which we've got a patent pending on, which is a swab collection tube. So you can take the swab, so take a nasopharyngeal swab, you put it into the tube, and once it's snapped off and sealed in the tube, nobody ever accesses that tube again. that tube is scanned and put straight into the machine. And the machine can sample from the same tube that it was transported in, and it's got the transport buffer in there, and that's how you release the RNA from the viral sample. So once again, completely without human intervention. Initially, that would be dedicated to COVID-19. The plan is in the longer term, we may make it available that you could run flu tests or something on it. But the initial short-term requirement would be to answer for a COVID-19 test.
I don't know what the limitations on the requirements for those tests, but I imagine that there has to be some kind of limits of detection. Do you conform to whatever that specification is with this?
We do meet the limits of detection on the kits. And the reason we can guarantee that is because we are using an established kit from a manufacturer who's already got a CE marked kit. That's a European manufacturer, and they're producing a standard CE marked kit. But the advantage to this system is, one, no human intervention. The next advantage is the fact that it can be used relatively near patient. So currently, all of the big systems, you have to batch up the samples, ship them off, run them, and then take them back to, the results go back to the patient. With this system, because it's relatively low cost, it can be sited in a clinic or a hospital, and you can have multiple units. It can run 22 patient samples at a go. So it's not a single cartridge unit, which makes it very cost effective to run. And the test costs are about $20 per person. And that compares very favorably with the current test costs, which are about $50 per person.
And what is the analysis time from start to finish?
The analysis time from start to finish, the first run takes 2 hours because you've got an hour of preparation and an hour of qPCR run, but every subsequent run is 1 hour because it can be doing the sample preparation whilst it's doing the qPCR run. So once you've got the first set of 22 samples off, you're getting 22 patient results every hour.
Right.
And we would expect that to run, theoretically it could run 24/7, but it just needs to have the consumables replenished. So obviously you don't have to load more test kits.
The data analysis side of things, does it print out a report for the clinicians, or?
It will either print out a report or it will directly interface into the laboratory LIMS system. So you can have it directly upload an electronic report into the hospital system. So it's got both functionalities. And that is, as I say, taking a raw sample, and it does the RNA extraction, then takes an RNA sample, places it on the plate, runs a qPCR on there, which would be a 3-plex system. So it covers 3 markers of proteins, and then you would get the results out at the end. And it's obviously just a positive, negative result.
Excellent. Is this available now?
No, it's not. It's going to take 6 months to produce. So that's the realistic time frame, because we're using widely established components within it and creating some custom components. So working with a UK manufacturer, they can manufacture this in a 6-month time frame.
So you're looking really realistic the end of this year?
We're realistically talking the autumn of this year, fall of this year. So, and that will enable the NHS. Initially, we would launch it in the UK, but obviously we're open to worldwide orders, but that would enable the NHS. The NHS currently has a target of 100,000 tests a day, but by the autumn, they have an unmentioned target, which is 250,000 tests a day. And with this style of machine, they would be able to meet that number of tests.
Right.
And because they'd be doing them near patient, there's none of the transport or delays that are built into using the large systems. So, I mean, the direct competitors are obviously near patient are cartridge-based systems. Yeah. So the likes of Cepheid, QIAGEN, StatDX. or in a high throughput laboratory, Roche or Becton Dickinson.
Right.
But they are, you know, a Roche machine is probably $1 million. And we're talking about a machine which is going to retail for £50,000.
Okay, that's a significant difference then in price.
It is.
What about the running costs? Are they standard qPCR and standard RNA extraction?
It's using magnetic bead extraction, but it uses a rapid magnetic bead extraction.
Okay.
And it uses our innovative consumable, which is an aluminium plate. So it's an aluminium plate which is covered in polypropylene. So that enables us to run it very fast. So we're talking about ramp rates on the qPCR of about 10 degrees per second, comparing that to a normal fast qPCR system, which is about 3 degrees per second.
Right. But so the product's developed or still in development? You've got a lot of information there on the product, which suggests it's pretty much developed. It's just a manufacturing.
It's developed. The research and development has been done on it. It's just the manufacturing which now needs to take place.
So I guess there's prototypes that have been built that give the proof of concept.
There's proof of concept, yes, and we have some prototype models. We don't have a fully working prototype model at this moment in time. But as I say, a lot of the established components within that come from established manufacturers, and we can build that into the system.
Right. Okay.
And it comes in a completely sealed housing. But
right.
One of the real beauties of it is you don't need to have qualified staff to run it. So it's literally put the tube in, press go, and it runs.
Okay. Sounds interesting.
Yeah, it sounds quite exciting.
Yeah. I mean, but today, the challenge of today, if we look at COVID-19, it's real-time PCR is
yes.
the detection method. And you have a system for that. You have a distribution channel for that. Can you just say a little bit about that product? Is it suitable for COVID-19 testing?
Well, the existing machine, which is the Express NGX, that's a research machine. So okay. We've never ever taken that to CE-IVD classification. So we know of a couple of groups worldwide that are using it for research, but it's not a clinically validated machine.
Right.
And we wouldn't really see that as a viable route, because having done the research, there are so many errors that creep in. And a lot of the work that's being done currently, you see they're incredibly manual. People are doing the RNA extraction in one machine. They then take a sample out, pipette it into another machine, seal it, run it, and it's all done in manual stages. Okay. What we've tried to do is pull together everything into one automated solution. And I've seen some papers which actually quote that the error rate is something like about 20% if you have human intervention in diagnostics tests. Right. So, I mean, 1 in 5 is a significant level of error.
Yeah, but today people have to work with the technologies that are available.
That's right. That's all there is available currently, and they're struggling. You know, they've got what they've got, and they're working well with it. But you know, there is the potential, you know, to do it much better.
But on your rapid real-time PCR machine, what are the and when you call it rapid, what do you mean by rapid relative to traditional?
Well, in the normal qPCR system, we can run a 40-cycle PCR in 15 minutes. Okay. And that compares favorably with a Bio-Rad system, which will take 50 minutes, and an ABI system, which may take just over an hour.
Even the fast system from ABI?
Yeah, it's that level of difference. Okay. And the reason is because we've got very accurate control over the temperature due to the fact that we're using an aluminium plate, and also we have a very, very thin layer of polypropylene on top of the aluminium. So we have 10 microns of polypropylene. So the only thing that separates the sample from the heat source is 10 microns of polypropylene. So in a conventional system, you have about 200 microns of polypropylene in the PCR plate. Then you have an air space. Then you have the silver heater block. And most of the existing systems will only do they'll use an algorithm to calculate the temperature that the qPCR is running at. With ours, it's a direct infrared measurement onto the bottom of the aluminium.
And your rapid instrument, that will run any PCR kit, or do you have to make adjustments to a kit that you buy?
You can run any kit on it, but often you have to do a period of validation because we're running it so fast.
Yeah.
Sometimes the hold times differ. And actually, one of the really significant things we notice is you have to change the hold temperatures, because people are convinced they're holding at 62, when in actual fact, they're probably holding at 60. So because ours is so accurate, if you set 62, it'll hold at 62.
Right.
It won't hold at 60. So you do need to validate the kit onto the system.
Okay. And this fast real-time PCR instrument you have today, this is part of the clinical instrument that you're developing, isn't it? That's
it's the same control body would go into it. Yes.
Same technology.
Yeah. The research machine that we sell has a complete onboard computer. It has complete support system, mechanical clamps, etc. The system that would be in the molecular diagnostic system is really a cut down, almost like a dumb black box,
right, that
sits inside the liquid handling solution and is controlled by an external computer.
Right. Ah, interesting. So on the new instrument,
yeah,
you mentioned some pretty big companies there that you're going to compete with, or that you're, where you will be competing in that market. What challenges do you expect?
Well, I think there are going to be challenges, but I think they're clearly defined into the areas they're working in. So if you've got a cartridge system, you're committed to a cartridge system. And the disadvantage with a cartridge system is there is an absolute cost to producing a cartridge.
Mm-hmm.
And you can run multiple cartridges at the same time, but in effect, it's a single user sample. It's one sample per cartridge. And what we've recognized is the fact that in that cartridge, you're throwing away your liquid handling system every time. If you look at the videos,
you're throwing it away.
Well, if you look at the videos that explain how these cartridges work, they're really, really complex set of liquid handling within the cartridge. And once the cartridge has been run, it's finished, you throw it away. That's why it has a higher cost. With ours, because it's using pipette tips and a liquid handling system, you're making the most of the liquid handling system, because all you're disposing of is a low value tip each time. You're not actually disposing of the complete liquid handling system.
Okay.
Obviously, if you compare it with the likes of Roche in the high throughput laboratories, they can deal with many more samples than we can, because as I say, we're only dealing 22 samples at a time.
Right.
But they are centralized, and within most countries there aren't that many centralized big laboratories that can A, afford one of these machines, or B, have the infrastructure to feed into it. So even for countries like America, where it's very disparate, it would make much more sense to be able to do the testing close to the patient, close to the hospital, rather than send it off to a central laboratory to run.
But in some healthcare systems, as I understand it, I'm not an expert, but where it's insurance-based, there are central laboratories that handle samples from those insurance companies' hospitals. So
yes, that's correct. That's correct. And I mean, but if you're dealing with something which needs an immediacy of results, there's got to be a way around that. There's got to be some clear thinking that says, hang on a minute, it doesn't make logical sense to wait until we've got 100 samples, put them in a van, drive them 60 miles, and then run a test which is going to take 4 hours, when we can do a local test and get the results off in 1 hour's time.
Yeah. Okay.
That's got some logic to it.
Do you have any, obviously, because of what's happened just now with COVID-19, you're targeting a different, or as you said, a simpler version of the instrument available later this year. Do you have customers for it now?
We've had various people phone up, and I mean, because obviously we're seen as being fast qPCR producers, people have phoned us to find out what we've got available, what we've got coming. So we haven't got customers sitting there waiting for us, because everybody's working immediately. And that, I think, is some of the shortfall, is the immediacy everybody's looking at. Whereas I think they need a bit of long-term planning in there as well. So, everybody says, oh, well, if it's going to take 6 months, we need something tomorrow. Right. Yes, but if you plan well and in 6 months' time you have a solution, that gives you a much greater strength, and it gives you a security, because there will be a second, third, whatever wave of COVID. It will keep going until vaccines or whatever are created. So that will carry on.
Now, for you with this instrument, your reagents are supplied by a partner. Is that correct?
Yes, that's correct. Yeah. We're not a reagent supplier. Right. We're purely an instrumentation research and development house.
But the instrument's a closed system, is it? Or can you, let's say you have, I don't need to name company names, but as we know, there are many different companies that supply RNA purification reagents, that supply qPCR reagents. Can people pick and choose, or?
In its initial form, it would be launched as a closed system.
Okay.
Because we'll optimize it to work exactly with the components we've put in.
Right.
Because that's one of the difficulties with this, is everybody will come along with a different test, and it can actually compromise the speed we can achieve, trying to fit in different people's foibles, or my kit needs this, it needs shaking, it needs aspirating. Whereas if we stick it to a closed system, then we can get a really clear run at it, and we know exactly that the system's reproducible.
Okay, right. Just
can I give you the website?
Well, I was just going to come to you just before we get to that. Is I think you've gone through everything on that system, which sounds, for me, it sounds pretty interesting. One question would be is, when it's available, how do you sell it? Do you use distributors, or do people buy direct from you?
Currently, we go through a distribution network. I think this is the sort of thing that a diagnostic company would be involved in. So I think we'd have to partner up with a diagnostics company that has international reach, because we are too small to do it. We don't have bodies on the ground to do the sales direct. Okay. So I think what we would do is partner up with somebody who would be able to provide us worldwide coverage.
Right. So you're looking for one partner, or you're open to using regional distributors?
Regional distributors is an option, but I think one big partner would be a better solution.
Okay.
Just because that would give us consistency of supply and a training issue. Obviously you train one group of people. Yeah. I know there's merit to using local distributors. We do that with our research and development machine, the one that goes into labs. Yeah. And there's some very, very strong relationships distributors have. Yeah. But I think with diagnostics, it almost has to come from a tier 1 supplier.
Okay, good. Then where can people find out more information? Okay. Now, what I'll do is when this goes on the website, I'll put the link under the video as well, but you can tell people now whoever's watching this, they can find out from you.
Okay, well, it's www.fastmdx.com.
www.fastmdx.com, and that will give them information about this new system that you talked about, yeah?
That will give them the full system, and you can see a video on there which runs.
Okay, and then for the research instrument?
That is www.bjsbio.com.
Okay, www.bjsbio.com.
Yeah.
All right. Okay then, Ralph. Wonderful. Thanks very much for your time. It was very interesting. I loved it. Thanks.
Thank you very much,
Okay.
Alistair.
Take
Bye.
care. Bye. Cheers.